Hi, so I'm very pleased to introduce our next speaker, Professor Stephen Walker. He's a professor of regenerative medicine at Wake Forest Institute for Regenerative Medicine. His group is focused on the application of integrated multi-omics approaches using specimens from patients to understand the molecular basis of chronic GI symptoms in children with autism, and to understand how these chronic GI symptoms early in childhood can affect neurodevelopment. So the objective of this work is to identify clinically relevant biomarkers that can be used for diagnosis or as therapeutic targets to the benefit of children with autism. So please join me in welcoming Dr. Walker.
Okay, can you hear me okay?
You're good.
Yep. Great. Okay. All right. Well, I want to start by thanking the Brain Foundation for inviting me to share our work with you. And good morning or good afternoon, depending on where you are. Dr. Krigsman just gave a really nice sort of overview of some of the molecular work we've undertaken for the last several years now. We're starting to move in further directions, I guess, as the field continues to grow. And so what I'm going to share with you today is some of the newer studies, or particularly a new pilot study that we've just undertaken. And the thesis of the concept is that microbiome analyses in autism and pretty much in all disorders right now is largely being done at the level of the fecal microbiome. And so we would posit that you might get more relevant or detailed information if you look actually at specific anatomic locations, for example, within the gut. And the usefulness of that may be to inform clinical decision-making, but we'll leave that open for questions at the end. So why are we interested in the microbiome? Well, like everyone, I think we can appreciate that what goes on in the gut, the body's largest immune system, populated by billions of bacteria and species, impacts the CNS and the brain, and it's a bidirectional relationship. So if you have alterations or a diseased gut, if you look at the image here on the right side, a diseased gut, you might see aberrant proliferation of stem cells that are meant to repair damage to the mucosa, for example. You might see altered neurotransmission, altered gut motility, which Dr. Krigsman spoke about just in the last talk, and a dysbiosis-induced change in the mucus layer. And I think we've heard from 2 talks ago that could have devastating impact on the permeability or impact on the permeability characteristics of the gut, allowing perhaps things to get through and out of the gut into the bloodstream that were not meant to do that. And so, again, the gut-brain axis, I think, has been reasonably well established. And this is just an image to give you an idea. There are a lot of things coming out of the gut that impact human physiology. And I think you could refer to a review for more information there. So the research strategies to evaluate microbiome in samples have taken on many forms. In column A here, pretty much the standard form, at least in the papers I've been reviewing lately. You typically have, in humans, you typically have a control group and some patient group, and typically the sample of choice is stool, and then you can isolate biomaterial from stool and look at the metabolomic profile, and that could be the metabolomic profile of the bacteria and or the host. And you could look at the fecal microbiota as to the diversity and abundance. There are a number of other approaches. You could look at, so starting in B, you could look at, starting with humans, isolate the healthy humans, isolate the microbiome from stool, transplant that into germ-free rodent models, and then evaluate all sorts of things, molecular, behavioral, and so on. Fecal transplantation, I think you're going to hear about that in the next two talks, so I'll leave that for them. And then finally, dietary interventions. I think we're aware of dysbiosis is often treated using probiotic and prebiotic, and then you evaluate the treated patient to determine if that's had a positive impact. And so, again, approaches to study the microbiome and its impact on human biology can take on a top-down approach or a bottom-up approach. And what our lab uses is pretty much a bottom-up approach. And so on the left there is a cartoon of the gut. And we can, within the gut, if we have samples from the gut, we can look at metabolites, either host metabolites and or metabolites produced by the microbiota. We can look at the microbes themselves as to abundance, diversity, and I'll make the argument we can also look at anatomic location specific profiles. And we can also look at genes and proteins that are either host related, microbial related, or both. And using these types of omics technologies, and some would argue more is better, more information is better, you can begin to build a picture of what dysbiosis actually looks like on a physiologic level. And so again, the standard approach, two patient groups, or sorry, a control and a patient group, get a stool sample, isolate the material from that, and then do metabolomics or microbiomic studies. And what I'll do now is go over a couple of the most recent review articles that look at the microbiome in an ASD. This one doesn't look necessarily at those with GI symptomatic children, but just children with autism spectrum disorder. And so this was published in 2019. The background for the study, they found that differences in microbiota composition in children with ASD compared to their unaffected siblings and healthy controls has been reported in a lot of studies. And so this particular review looked at studies published between 1966 and 2019. That's a long window. Most don't go back that long. But anyway, they identified 28 studies that are the subject of the review article. And what they found was the patients in these studies range from between 12 to 104 participants, between 2 and 18 years old, and then from various geographical areas. And a majority of these studies did microbiome analysis through fecal samples. However, out of the 28, there were 4 studies that looked at mucosal biopsies from different sites within the GI tract. And I bring that up because I'm going to talk about those 4 studies in just a moment. What this review article concluded was the heterogeneity in all of these things, the way you diagnose ASD, the site of the gut that you sample or the stool sample that you use, the laboratory methods that you use, and so on, make a meta-analysis of a set of articles like this inappropriate because there are too many variables and you can't compare them directly. But the take-home message was each of those studies, or the majority of the studies, find that gut microbiome is altered in ASD, although they conclude further exploration is needed to determine whether or not this is a cause or effect. In other words, does having autism predispose you to having a dysbiotic gut, or is it the other way around? On to those 4 studies that looked at mucosal specimens, particularly because, again, that's an area that's of interest to us. The first study many of you would be familiar with is the Williams study published in 2011. And in this study, there were 22 participants, 15 with autism, 7 control. They were all about 4 years old on average, male children with GI dysfunction. The measurements that they took in this set of studies was disaccharidase and hexose transporter gene expression, i.e. mRNA, in the ileum. And then they also looked at bacterial 16S sequencing in the ileum and in fecal biopsies. So the first finding was the disaccharidase and hexose transporter transcript level, gene levels, were reduced in the autism samples with GI dysfunction. The second finding was that the autism GI children have a microbial dysbiosis, and they concluded that the dysbiosis is associated, or they showed that the dysbiosis is associated with the host deficiencies in these transcript levels. Interesting stuff. So a year later, 2012, Williams and a group of researchers, they had determined in the original 2011 study that Pseudorella was a species that popped up as maybe being especially interesting and important for this description or delineation between autism and control samples. So in this study, it's basically a molecular study where they developed a PCR-based test to detect and quantify the specific Pseudorella species. And so in this study, they had 23 autism children and 9 control, all male children with GI disturbances. They did a PCR-based method of determination of the Pseudorella species amount. And they found the Pseudorella was present in 12 of the 23, so about half of the autism GI samples, and none of the 9 controls, suggesting that this may be a biomarker for dysbiosis in at least some subset of GI symptomatic children with autism. The third study that used, that's cited in the early review that actually used biopsies to look at microbiome was from Ruth and Luna's group, published in 2016. In this study, they looked at children with ASD and functional gastrointestinal disorder. So 14 of those children, 15 neurotypical with also had functional gastrointestinal disorder, and then 6 neurotypical children without any FGID. And they looked at rectal biopsies, and they did a 16S, again, 16S RNA sequencing. And this was a much larger study. I'm distilling it down just to the microbiome findings. Distinct microbiome and cytokine measures in rectal mucosa of children with ASD and functional GI disorder compared to neurotypical with or without functional GID. So there was a difference in this case, maybe ascribed largely to autism. And then finally, a study by Kushak et al. And again, this is some of the authors here on the original 2 studies. I think you'll find in studies like this, oftentimes it's the same group or subgroups of people doing this work, because there aren't a lot of people doing this type of work at the present time. But anyway, they looked at 21 kids with autism and 19 unaffected kids undergoing upper endoscopy now. So now we're looking at the upper GI. They were all being scoped for suspected GI disorders. They took duodenal biopsies and analyzed disaccharidase activity and also 16S sequencing for bacterial species identification. They found in this case that there was no statistically significant difference in the microbiome diversity in the duodenum between autistic and non-autistic children with GI symptoms, but there was a positive correlation between the abundance of Clostridium species and disaccharidase activity in the autistic children alone. Okay, so the second and more current review, this was done in, this was published in 2020 in the journal Autism, it's the link between ASD and gut microbiota. And in this case, they report that GI distress is present in up to 90% of autism spectrum disorder cases. Again, these numbers, depending on the literature that you're reviewing, vary, but so this is a pretty high number. They looked only at studies conducted in the past 6 years that examined the association between gut microbiota and ASD, and they included 19 studies in their review. And what they found was the research continues to report differences between the microbiota of individuals with ASD and controls. However, the types and abundances of bacteria present remain inconsistent, or I might argue inconsistently reported. And there's good reasons for that. We'll get to that in the next slide. And they further conclude that rigorous methodologies are required to support and strengthen the reliability of these existing results. And I wouldn't disagree with that. They spend some time in the discussion talking about the need for subgrouping. And I will tell you from experience that when you're looking at a complex disorder, highly heterogeneous like autism is, or any of a number of other complex disorders, what you understand is there are a whole host of phenotypes, maybe subphenotypes even. And so it's really important if you're going to do a study of any type, for example, a clinical trial looking at efficacy. The reason a lot of clinical trials fail is because, for example, in autism, let's just say they chose 100 people with an autism diagnosis and applied some therapeutic, and then they looked at the efficacy of the therapeutic and they found it was only 20% efficacious. So you say, well, that drug's not very good, or that therapeutic's not very good. The real answer might be there are 20% of that 100 that have an etiology that's more similar than the other 80%. And therefore that therapeutic is absolutely applicable to that subset. And so if you were to define the subset or describe a subset of patients' characteristics beforehand, and likewise with research studies, you would probably end up with information that was more, number one, useful, and number two, comparable across studies. And so that's just a long-winded way of saying what I've written there on the slide. So the need for subgrouping, I think, is important, and there are any of a number of reasons why, but the condition's heterogeneous, the exposures are heterogeneous, the drugs are heterogeneous, and some of those things are more important than others, but it's important to take note of those things like antibiotic use and so forth if you're looking at a microbiome analysis. Okay, and as I was doing this literature review in the last week or so, I came across this paper that I thought I'd throw in here. If you're interested in this topic, I think I'm always one to pitch for standardization. And what they're arguing here is that if you want to understand dysbiosis, a precursor step that's really significant and important is to understand what a normal, normal in quotes, microbiome in a healthy population looks like. And it's going to be variable and heterogeneous, but you should be able to come up with a consistent sort of minimal profile, if you will. And we did this back when I was doing blood gene expression profiling early on. We had the same issue. If you want to know if the genes or proteins are dysregulated in blood, it's really important to know what a standard or a typical profile would look like in whatever population or group that you're looking at, whether it's a pediatric or adult. You need to know what the baseline or the standard looks like in order to know if what you're looking at is irregular. Now, there are some exceptions, of course. You can identify disease biomarkers sometimes just on their face, but the point is it's good to have a baseline of what so-called normal to work from. Anyway, so our pilot study, the question that we set out to answer with this pilot study is the fecal microbiome a good proxy for the mucosal microbiome? And the hypotheses that we would put forth are that the mucosal, so the mucosal would be, multiple locations within the gut. The mucosal microbiome composition will vary by anatomic location within the gut. And I think that's reasonable to assume. The mucosal microbiome will differ significantly from the fecal microbiome. I guess I think of the fecal microbiome as sort of the composite biome of everything throughout the gut in some way. And then finally, the mucosal microbiome will differ significantly from those with autism and those without autism who have gastrointestinal symptoms. And so our study group for this really small pilot is 5 GI symptomatic male children who had either a diagnosis of ASD or developmental delay, who were undergoing clinically indicated endoscopy. And the biopsy collection was from multiple sites in the lower GI tract. I've indicated 5 in this image. For this study, we took biopsies from 3 of those 5 sites to be representative of the whole colon. So the terminal ileum, the transverse colon, and rectum. And the methods that we used were that and this is interesting, I wasn't aware of this, and I'll get to why this is important at the end of the talk, but I wasn't aware several years ago that you could actually do a microbiome analysis on pinch biopsies that had been stored in RNA later. And we have thousands upon thousands of those. And when I found out that's not only, you're not only able to do it, but you're able to do it at a relatively reasonable expense, it opens up a lot of possibilities for things we can do, including this pilot study. So we extract microbial DNA and RNA from the biopsy samples. We perform, in this case, shotgun metagenomic sequencing. So this is a comprehensive sequencing rather than a 16S rRNA sequencing. That's specific for the bacterial species. The shotgun metagenomic sequences everything, and then you match it against, you match the reads against reference databases, and then you can identify what's bacteria, what's fungi, what's virus, and what's host. And by doing this, number one, I mean, people do metagenomic sequencing and only use the bacterial data to report, but you have all that other information. You can report on all of these things if you do metagenomic sequencing with deep enough read depth. Anyway, at this point, then you can identify and quantify microorganisms in each sample. So that's what we did. So in these 5 patients, what you're looking at here is the microbial diversity in the 5 patients that I described. So across the top is patient 1 through 5. Across the bottom, it's terminal ileum, transverse colon, and rectum, same order all the way across. And what you can see without even really knowing what you're looking at maybe is that within an individual, the profiles are quite different at the 3 sites. And across individuals, for example, look at individual number 3 and compare that to individual number 5. There's quite a bit of difference there, even if you don't know at this point exactly what bacterial species you're looking at. You can see from this that there's quite a bit of difference. If you sum all the terminal ileum profiles, the transverse colon abundance profiles, and the rectum abundance profiles, now you get a sort of an average microbial diversity at each site. And you can see there appears to be less diversity at the terminal ileum than either the colon, the transverse colon, or the rectum. And if you look at this in a principal component analysis plot, what you can notice immediately is the 5 dark blue dots in the center of this PCA plot are the terminal ileum profiles, and they group very closely together, which is what you saw in the previous bar graph. The other 2 sets from transverse colon and rectum are much more widely distributed within this PCA plot. And what that tells you, without even seeing the bar graphs in the previous slide, is that these samples, there's a lot of individual variability in these samples. And that's important. It doesn't mean one is ill and the other isn't, but it means there's a lot of individual variability that has to be reckoned with in your analysis. So the findings within our little pilot study, and thinking back to the hypotheses that we were seeking to address, between anatomic locations, and the microbial composition in the TI was less diverse and more similar across the 5 individuals compared to transverse colon and rectum. So we have a large biorepository. By we, I mean Dr. Krigsman and I have been accumulating this biorepository from his patients that he's been treating now for many years. So for ASD children, we have samples from 1,320 single encounters, so 1,320 individuals and their clinical information. We have 154 sets, and I'll tell you what the sets encompass in just a moment, that had 2 encounters, meaning they had 2 endoscopies, and so we have samples from both of those, and then some with even 3 and 4 procedures. And then the typically developing controlled children, and to understand these typically developing controlled children are all had GI symptoms and they went in for endoscopy. Otherwise, we wouldn't be getting GI samples if they didn't. And so we have at least 400 sample sets from those. And a sample set in this biorepository consists of a large number of samples. So for the ASD children, each time Dr. Krigsman scopes one of the children, we get 12 to 15 GI biopsies from throughout the upper and lower GI. And these are stored in a preservative called RNA later, which allows us to later do now microbiome work, but RNA or gene expression work. A couple 2 to 4 flash frozen biopsies are taken at locations like the terminal ileum, duodenum, and cecum. This will allow us to do other work like protein work and so forth that you can't do with samples stored in RNA later. We have whole blood in these special tubes that preserve the integrity of the whole blood, and we're able to do gene expression work with that. And then we have serum and plasma that we can do metabolite, peripheral metabolite studies. This is on every single patient that I showed you before. So 1,506 sets times 30 or 40 samples per set. That's a lot of biosamples. And then for the controls, Not quite as exhaustive, but we have 6 to 12 biopsies and RNA later from each case, whole blood and packaging tubes, serum and plasma. So this is our comparator group in most cases. And so the next steps, we would like to develop an in vitro model to model the gut of a child with a GI symptomatic child with autism. So there's a couple ways to do that. You can take an intestinal biopsy or a tissue resection from a patient, and then you can do some cell culture and then do multi-omic analysis on the samples. Another way to do it is with pluripotent stem cells. And we've actually done work in both of these arenas. But what I'll focus on right now is our most recent work, and this is just a one slide thing, on intestinal biopsy modeling using intestinal biopsy. So what we're creating here is gut organoids. And if you saw the talks last week, you heard Paul Ashwood talk about this a little bit. So basically, when a patient undergoes a colonoscopy, we take the biopsy samples, as I indicated, and we have thousands of these samples. These need to be taken fresh. So you take the fresh biopsy sample, you isolate the crypts from the mucosal biopsy, and this can be, from anywhere you like, the TI, the cecum, the rectum, anywhere. And you, in cell culture, create then these epithelial mini gut organoids. And there's all sorts of work being done in this. We haven't done this yet. We haven't started the modeling yet. We just started making the organoids just to see if we could, see how difficult it is. And there are a ton of things we can do to evaluate a patient-specific GI model, if you will. And so in simplistic form, it's just the intestinal crypts are isolated from the biopsy tissue. They are then grown in three-dimensional culture, and they self-assemble to create a lumen, and they differentiate to create each of the cell types listed to the right there that comprise the lumen. And so there are other things that you can do with this. Once you have these gut organoids, you can turn them inside out. You can apply your own microbiome, if you like. Lots of things we can do to evaluate this. And the cool thing is that these model systems will be patient specific. So they will theoretically model, if the patient has a leaky gut, for example, that may be recapitulated in a gut organoid model. And those are the types of things we would like to look at. And so our first foray into this, we literally just started not more than a week ago. We got fresh tissue from a procedure that Dr. Krigsman did. We isolated the crypts and we created gut organoids. And so in the first two slides, you see what a gut organoid looks after growth about a week in media and matrigel. These are three-dimensional organoids. And then on the right is a healthy control. And you'll notice right away that the two images on the left have a lot of vacuolization and so forth. Don't know quite what that means yet, but they look quite different from the healthy control. And these are from an individual patient, so we can do this from a large number of patients at the end of the day. And so with that, I'll end, and I'd like to acknowledge my collaborators, Dr. Krigsman, for many, many years now. He provides the clinical specimens, the clinical expertise, friendship, and all the things that go with work like this. Dr. Reeves is in the Center for Precision Medicine here at Wake Forest, and she has performed the metagenomic analysis and will perform the metatranscriptomic and other analyses for us. Dr. Bishop is an expert in gut organoids. He's here at the Institute for Regenerative Medicine, and he's going to work with me on model development. And then finally, the Wake Forest Institute for Regen Medicine Organoid Core Facility. And with that, I'll end.
Okay, thank you very much, Steve. Great presentation. We have one quick question for you. Does the microbial population change dynamically with time? And is there therefore value in sequencing past data except for longitudinal observation for comparison? And do we need to match by age?
Boy, well, that's a couple of questions. Number one, I'm told that the biome in the colonic biome doesn't change turnover as quickly, if you will, as in the upper GI. The biome there apparently turns over much more quickly. So the answer to that is, I don't think that the turnover is as rapid in the lower GI. So if you do a sample that you took this year and a sample that you took next year, in terms of the utility of that, you know, it depends. If there are differences and you also have differences in the clinical picture, then it may be related. I mean, we haven't done that yet, so I don't know the answer. In fact, you guys in the next two talks may be able to address that more directly. I'm sorry, what was the last part of the question?
And when you do your matching, you try to match by age?
I think it's critically important. Again, alluding back to the review articles that I talked about, I mean, it's very important to narrow the amount of differences between your study populations as best you can, and age is, I think, going to be an important factor, but not the only one, of course.
Okay, and then one other question, would it be feasible to have a project with gut and brain organoids from the same patient cohorts and compare them?
Oh, I'm glad you asked. So a proposal that we put forward building this gut model is exactly to do that. We would do that with induced pluripotent stem cells because we are not getting, obviously, brain biopsies from these patients, and you don't need gut biopsies either. In other words, you could take a skin cell or a blood cell even from a specific patient, and we have proposed this, yes. You can then take the cells, bring them back to a pluripotent state, and take them down a gut pathway, and take another set of the cells through a brain pathway. And then in a microfluidic device, you could attach those two by media, which would serve as the conduit between the gut and the brain, and look at all sorts of things. So the answer is absolutely.